to discuss about immunity and related disorders. So let's get started. First of all, you need to know what does antigen mean. Antigen is any foreign substance or any substance inside the body that can stimulate immune response. So it can be foreign or it can be inside the body. Alright, having known this, let's need to know what does hapten mean. Hapten is any molecule like antigen, but it is composed of a protein structure. So it is composed of protein structure. Alright, now we will discuss immunity, two types of immunity, cellular and humoral. First, I'll tell you about cell-mediated immunity. The cell-mediated immunity is mainly mediated by the T lymphocytes. So first of all, the antigen comes inside the body and this antigen will be captured and processed. By macrophage. The processed antigen will be exposed on the cell membrane of a T cell receptor and it will be recognized by the T helper cell. The T helper cell will recognize the antigen. Now that it has recognized the antigen, it will stimulate the T cells, T killer cells. The T killer cells will be stimulated or given signals so as to encounter the antigen. Alright, so the T cell will start to signal lymphokines. As a result, these lymphokines will stimulate inflammatory reaction at the site of antigenic invasion. So there is inflammatory invasion. Alright, so by this the antigen is destroyed inside the body. So there are some certain cells that which is stopped at G1 phase. It is called as memory cell. These memory cells are activated during the secondary immune response, like when the antigen comes in, the whole process from first to fifth begins and the memory cell will recognize the antigen and keep it as an information, as a memory, so that when it comes secondarily, it can stimulate the immune response against it. So the secondary immune response is much more efficient than primary. So this is humoral immunity is mainly mediated by B lymphocyte. But certain other cells like A cell and T lymphocyte also play their own roles. So let's see what happens. So in the humoral immunity, the steps over here is the same from the antigenic invasion to the capturing and processing and the recognition of T cell, T helper cell. Alright, after this what happens is what makes it different. So let me tell you over here. what happens next? The T helper cell will stimulate the B lymphocytes. Now, the B lymphocytes got stimulated. After its stimulation, what happens is the B lymphocytes will produce antibodies. How will it produce antibodies? It will produce antibodies with the proliferation of plasma cells. But before that, what happens is exactly I'll tell you. The B cells or the B lymphocytes will undergo biotransformation. As a result of biotransformation, plasmablasts are formed. These plasmablasts undergo clonal proliferation. Let's make it this next point. Further clonal proliferation starts of plasmablast, and they will form plasma cells or plasmocytes. Now these plasma cells will produce specific antibodies. In this video, I'll discuss about the types of antibodies. Types of antibodies are of 5 types. First one is IgA. The IgA is mainly present in tears, saliva, and colostrum. Most of you might have known what does colostrum mean. It is the first form of milk which is produced by mammals. That is, for example, you can take it as humans. Next type is IgD. The exact location of IgD is still unknown. Alright, next, the third one is IgE. IgE is only present in the mammals, and it mediates allergic responses in allergic reactions. I'll tell you about the hypersensitivity reactions a little later. So this IgE is mediated first hypersensitivity reactions. It is very important to be noted. Hypersensitivity. Alright. The next type is IgG. IgG. Alright. This IgG has further 4 subclasses. It will provide long-term defense from antigen. It can pass through the placenta, providing immunity to the fetus. So this point is very important to be noted, that IgG antibody can pass through the placenta and it provides immunity to the fetus. Alright, next, moving on to the fifth one, that is IgM. This IgM is the largest antibody which is synthesized during the primary immune response. So, as I told you about over here, the primary immune response, whatever is elicited in the humoral immunity, the antibodies are produced. So, the main antibodies produced in the humoral immunity seems to be IgM and IgG. So, I'll tell you about this mechanism in a moment. So first, IgM is the largest antibody. It is the largest antibody synthesized during the primary immune response. Alright, primary immunity. The IgG and IgM M antibodies will eliminate the alien antigen or foreign antigen by the process known as opsonization. Eliminate the antigen by a process known as opsonization. In this opsonization, the antigen is coated by antibody and taken up by a phagocyte or NK cell. So it is destroyed by phagocyte and NK cell. So when its concentration, when these both of the antibodies concentration in the body increases, it is limited by another type of T lymphocyte that is called as T suppressor. So the T suppressor is another name, another type of T lymphocyte. So we have got totally three types of T lymphocytes. They are T helper cells. They are also called as CD4 plus cells. T killer cells. They are also called as CD8 plus cells. And now we have got T suppressor cell. This cell will suppress the IgG and IgM. Alright, having said this, now we will go to the disorders. Primary immune deficiencies, and we will conclude it. The primary immune deficiencies, first of all, we will look at humoral immunity deficiency. Immunity deficiency. The primary immunodeficiencies are also called as congenital immune deficiencies. Alright, so in the humoral immunity deficiency, there is a specific name of this disorder called as X-linked A gamma globulinemia. There is one more name for this type of disorder that is called as Bruton's A gamma globulinemia. Here, let us see how it is characterized by. It is characterized by decrease or absence of B cells. So these points are really very important to be noted. There is decrease or complete absence of B cells or B lymphocytes, and also class of immunoglobulins. Class of immunoglobulin G, IgG, IgM, IgA may also be deficit. One more clinical feature here presents to be the germinal center of lymph nodes, Peyer's patches, appendix, tonsils are underdeveloped. These all are underdeveloped. Alright. The T cell over here will be absolutely normal. T cell presents to be normal. For example, the example of this type of disorder is tend to be pneumonia, in cases of pneumonia, bronchitis, pharyngitis, etc. The important point over here you need to know is that there is deficiency or complete absence of B lymphocyte. This is what you have to understand over here. Alright. The next point is cellular immunity. The second one is cellular immunity deficiency or disorder. So in this, the particular name of this disorder is Digeorge Syndrome. It's called as Digeorge Syndrome. As a result, there will be decrease of or hypoplasia of thymus. Parathyroid glands may also be affected. So I'll let you know over here. There is congenital deficiency of third and fourth pharyngeal pouches. So as a result of this deficiency, there is thymus hypoplasia. If there is thymus hypoplasia, the number of T cells are proliferated and got matured in the thymus. So if there is hypoplasia of thymus, even the T lymphocytes number will decrease. So the T cells are decreased or absent in number. So it is caused over here. Let me write it over here. Thymus hypoplasia. Alright. So the next one, the examples of this type of deficiency is it occurs in viral, fungal, protozoal infections. Alright. The B cell and the serum immunoglobulin are entirely unaffected. B cells and serum immunoglobulins are entirely unaffected. Alright. Now we will move on to the third type of immunodeficiency, that is called as Swiss type A gamma globulinemia. Swiss type A gamma globulinemia. This is also called as severe combined immunodeficiency disease. Severe combined immunodeficiency. So this is an X-linked disorder. In this type of immunodeficiency, there is B and T. Both of the lymphocytes are lacking or absent. The same features, thymus is hypoplastic over here. The thymus is hypoplastic and the lymph nodes are very hard to find. Like the lymph nodes of the body are decreased in number. The infants are vulnerable to all forms of viral, fungal and bacterial infection. So congenital means what? It occurs in the infants, right? So moving on further to the next type of immunodeficiency, that is Lewis-Barre syndrome. In the Lewis-Barre syndrome, this syndrome is also called as ataxia telangiectasia. Ataxia, that means poor coordination, and telangiectasia. Telangiectasia means the veins of the body will get blood vessels will get dilated and it will appear as a spider. So it is called a spider angioma. This syndrome is also called as ataxia telangiectasia disorder or disease. Alright. These are the findings of this type of syndrome, and later, since I told that Lewis-Barre syndrome is accompanied by decrease of T lymphocytes and immunoglobulins, there is decrease of T lymphocyte and immunoglobulin A, immunoglobulin G, immunoglobulin M. All of these will be deficient in this type of syndrome. The upper and lower, it is followed by upper and lower respiratory tract infections. Upper and lower respiratory tract infections are very common in this case. Alright, moving on further to another type of syndrome, that is known as Wiskott-Aldrich syndrome. Wiskott-Aldrich syndrome. It is also X-linked disorder, recessive disorder. Here we can find IgM and T lymphocyte. IgM combined with T lymphocyte deficiency. This syndrome is also called as eczema thrombocytopenia. Eczema means rashes type on the skin, and thrombocytopenia means decrease of platelet number. Thrombocytopenia. Alright. Another type of disorder is called as phagocyte disorder. It is given phagocyte. Phagocytes are what? Macrophages, dendritic cells, any of them. So as a result of phagocyte disorder, there is given a specific name called as chronic granulomatosis. This is the specific name given for this. Another one is celiac type of syndrome is called as celiac Higashi syndrome. In the celiac Higashi syndrome, there is impaired white blood cell function due to failure of phagolysosome formation. There is white blood cell function defect due to phagolysosome failure. So these are the entire disorders of the immune system. So that's it for today. Thank you.